finish up our topic on ATP production and macromolecule catabolism, we're going to finish up with enzymatic activity, reaction rate regulation, and coupled reactions. And this kind of will help take a lot of the specifics that we had in our biochemical pathways and kind of look at it with a larger scope to get a general appreciation for what's truly going on. In this section, you should be able to understand what an enzyme is and what it's going to accomplish in the body. We're going to look at different models of function. And because enzymes are going to be so essential to reactions occurring in the body, we're going to look at how we can use these enzymatics and how we can control the rate of these various reactions. We also need to understand the coupling of reactions, energy-consuming reactions, energy-producing reactions. Now, we take those both of those types and couple them together to make the system run within the laws of our thermodynamics. This is going to be a general energetic diagram that you'll see pop up a little bit later. Basically, we have energy up the vertical axis and time across the bottom. So the line within that graph is going to represent the various energy levels at points in time as a reaction progresses. Now, we're going to be mostly concerned with the start and the finish point, as you'll see when we get done today. Are we starting at a higher energy level than when we're finishing? If so, energy had to be expelled. We don't just lose energy, it had to go somewhere. As opposed to if we're starting off at a low energy state and producing products that are at a higher energy state, therefore implying we had to put energy into that system. But for starters, I want to draw our attention to this portion. When we start a reaction and have to go to a higher energetic intermediate state before progressing to an ultimately lower energetic state product. This bump in the road, so to speak, is called the activation energy. And it's going to be the catalyst job, or the enzyme's job, to get us over the hump. Now, I mentioned the word catalyst there for just a moment. You may have heard of that term before, if you've taken any chemistry classes. If you were doing a tabletop chemistry experiment, and you were wanting to progress it from reactants to products, different reactions may require a catalyst. It's going to be an ingredient into the reaction that helps that reaction take place quicker. What it essentially does is it lowers the activation energy. You may have a reaction that has to sit on top of a Bunsen burner, get to a certain temperature for such a long period of time in order for it to carry out to completion. But with the catalyst present, you may be able to get to completion at a lower temperature, or you may be able to get to completion in a much shorter period of time. It helps the reactants overcome the activation energy so it can progress to the product state. So if we're a tabletop chemistry reaction, we refer to that as a catalyst. In the human body, the catalysts that we have for reactions we talked about last class are going to be referred to as enzymes. These are the entities in charge with helping our various reactions that we've already talked about progress from a reactant to a product state. It's important to realize that catalysts and enzymes are not consumed by the reaction. Again, tabletop chemistry experiment. You have your reactant, you put your catalyst in, at the end of it you have your products. You can get 100% of the catalyst back out of that flask. They are not consumed in the reaction. They take part in the reaction, helping it progress, but they are not consumed by the reaction. An analogy I've used for this for a while is workers in a factory. The workers are catalysts, helping spare parts go to a finished product. However, the worker is not consumed in that reaction. At the end of the day, the worker is still able to come back and work on more spare parts over and over and over again. So just like a catalyst, enzymes are not consumed. You eat a large macromolecule of carbohydrate. We mentioned salivary and pancreatic amylase. Those are enzymes. They bind to that large macromolecule, they start chipping pieces off the end, breaking it down. But they're not consumed in that reaction. They can continue to act as long as their protein structure integrity is intact. Enzymes are going to require very specific ranges of their environment in order to maintain optimal function. Now, this should sound at least something like the dynamic consistency that we've described for maintaining homeostasis. Every enzyme has a set of optimal environmental conditions at which it works best. Now, if we go back one slide, you may already start to get an appreciation for if the enzyme has a great responsibility in helping a reaction go from reactants to products, and we can change its optimal ability in which it does that function, then we should be able to control the rate at which a reaction takes place by manipulating the efficiency of the enzyme. This is going to be our gas pedal and our brake pedal when it comes to controlling how quickly reactions take place. Every reaction we've talked about so far, there are going to be times when we want it to progress more than not, or vice versa. We manipulate the various enzymes' activity to control the rate of reaction. Let's look at a couple of scenarios where we can really wrap our minds around it more easily, where we can see enzymes and their efficiency changing. Here's temperature versus enzyme activity. What do you suppose is the temperature that most of the enzymes function best in the human body? 98.6, typical normal body temperature. And you see this somewhat of a bell curve. You start getting into a situation where the temperature is escalating. 101, 102, 103, 104. The further away from the optimal body temperature, the less efficient various enzymes are going to become. Much tougher to do their job, therefore not able to progress reactions as they should be. Same goes for if we were to drop you into the Atlantic Ocean here now, North Atlantic. Hypothermia sets in, body temperature starts to plummet, enzyme activity goes down. By changing the temperature of the body, we can move the enzymes outside of their most effective environment, therefore decreasing their efficiency at which they can operate. We mentioned it last time about salivary amylase. How when we're in the saliva in the mouth, somewhere at a pH of just short of 7, it works okay. Now, by that point, we haven't done much physical breakdown yet because we just started chewing our food, but that's where it works well, in the pH of the mouth. You take that salivary amylase and you secrete it into the food you just chewed up, and then you swallow it. And you put it in the stomach, where now the pH is just less than 3, or just around 3. We are well outside the optimal range for salivary amylase. It becomes completely ineffective once it hits the environment of the stomach contents. So we manipulate the environment, we can change the efficiency, therefore we can change the rate of reaction. Now, are we going to want to use body temperature as a way of controlling body reactions? Of course not. These are not going to be viable means of adequately controlling reaction rates. They're not specific enough. We do have a couple of means by which we can control enzyme activity, though, that doesn't involve putting the body in a complete state of non-viability. The first way that we have to control enzyme activity that we'll discuss, and it's probably going to be the most effective way, is that we'll talk about 4. This is the biggie. End product inhibition. is the most valuable means of control we have over enzymes' activity, therefore the most valuable way that we have to control reaction rates. It basically states that in a cascade of reactions, the product that pops up at the very end of that cascade inhibits enzyme activity further back in the chain. Now, we've looked at cascades of reactions before. As I'm saying that, you may be asking, what does he mean by a cascade of reactions? Think about glycolysis. We started with glucose at the top. We moved it to glucose 6-phosphate. We had 7 or 8 different steps until we ended up with pyruvate at the very end. This could be glucose, this could be glucose 6-phosphate, this could be pyruvate, and we had individual enzymes controlling each step of that way. So we've already discussed several reaction processes that occur as a cascade of individual reactions. But in either case, the end product inhibits the activity of enzymes further back in the chain. Now, we're not going to get into exactly the mechanism of which it does that. It's unnecessary for us at this point. Just as an example, maybe the end product over here, E, binds to enzyme 1. It ties it up and it keeps it from acting on A to convert it to B. Maybe in some situations, the end product may bind to the original reactant, keeping the enzyme from able to get to it. The mechanism is somewhat irrelevant, but it's the end product that makes this enzyme work less efficiently. It slows down the rate at which A can go to B. And if we've slowed down that initial one, then we can't produce C as readily, we can't produce D as readily, and we can't even produce more end product as readily. Now, as a result of this, you see this sinusoidal wave pattern as we look at how much E we ever have available at any one time. Because the more E we accumulate, the more inhibition we're going to exhibit. The less E we have, the less inhibition we can exhibit. So A goes to B, B goes to C as normal, and we start accumulating E. The more we get, the more inhibition we have. So E starts to climb, but then it starts going back and turning off the process. At that point, E will start to drop. The important thing to realize is that the end product in any chain of reactions is always going to be one of 2 broad categories. It's either going to be a waste product, which we're going to get rid of, or it's going to be something that we have a use for. There really isn't anything else that we can produce that won't fall in those 2 categories. Either we want it to use it for another purpose, or it's a waste product that we want to eliminate. Either way, if we stop producing it, the amount that we have at any given moment will start to decline. So we let our reaction cascade run. We see our end product accumulate. We shut down production. Now the levels of that end product will start to decline. Once they dip down to a certain point, inhibition will be released, and we will start to produce again. So we will see this accumulation and depletion pattern cyclically with respect to our end product as time goes on because of the cyclical inhibition that it's going to be applying further back in the chain. This is going to be a good place for you to really start focusing on one concept, the concept of active inhibition. We will touch on that particular term a lot once we get to neuro. The thought that we can do something actively and it stops something else from occurring. When you get done listening to this lecture and you go and you get in your car, you can actively slow your car down by actively putting on the brakes. Or you can passively slow your car down by simply taking your foot off the gas. Both of those actions will result in a stopped car. Here we have the first example of active inhibition. We've got an end product purposefully ceasing the actions of another enzyme. Now, we have 4 of these mechanisms of control to discuss. I said this was the most important one. Not only is it the most important one, the next 2 doesn't even really exist. They're more theoretical. But let's discuss them anyway to get a full scope of what we're talking about. The second mechanism is what we're going to call substrate depletion. And this is less of a mechanism, more of an observable phenomenon. A substrate, as you'll see in a moment, is what the enzyme acts on. In my analogy earlier of factory workers with spare parts, the spare parts are the substrate. The enzyme workers act on the substrate spare parts. The fewer spare parts we have, the less work is going to get done. And that's just an observable phenomenon. If we had 50 workers in a factory and an abundance of raw materials, we would see 100% efficiency. Every worker would have something to do. If we only had enough raw materials for half the workers, then we would get to a 50% efficiency. If we only had enough raw materials for 20% of the workers, then we have a 20% efficiency. And if we have no raw materials at all, we're going to have 50 workers standing around twiddling their thumbs with nothing to do. If there is no substrate, there can't be enzyme activity because there's nothing for the enzyme to act on. So we won't be considering this so much of a mechanism of control, but more so an observable phenomenon. Number 3, product accumulation. So we had substrate depletion, now we got product accumulation, and they sound like they're 2 sides of the same coin. And just by the name, I guess they would be, but the phenomena observed is not 2 sides of the same coin. This touches on the fact that we can have equilibrium. A plus B going to C, and of course we got that bidirectional arrow, means that C can just as readily break down to B and A. The reaction can take place in the forward or reverse directions readily. The problem with this being a means of communication, or a means of regulation, goes back to somewhat of the fatal flaw we had early on when we categorize what E is. It's either something we want to use in the body, consume it, or it's something we want to get rid of. Either way, it never lingers. So if we are going from reactants to products here, and we've got an enzyme handling that transition, if we keep A and B quantities stable, and we let C just sit around, then yes, we'll reach an equilibrium. And at that point, enzyme activity will go to 0 because there's nothing for it to do. We're accomplishing nothing. The reaction is taking place at equal rates forward and backward. But it's assuming that we're allowing these particular entities to just linger. And because of our 2 broad categories of something either being useful or a waste product, that doesn't exactly occur. And the 4th type, now this is a legitimate mechanism of control. It's a little bit more abstract than what we've talked about so far, though. The concept of genetic control. Now for this, we're going to go back and use my analogy of you stopping your car again. Genes can be turned on or turned off, induced or repressed. Depending on what the product of that gene is, will determine if induction or repression increases enzyme activity or represses enzyme activity. Let's talk about making your car stop again. If you induce the gas pedal, it's going to accelerate. If you induce the brake pedal, it's going to decelerate. If you repress the gas pedal, take your foot off the gas, it's going to decelerate. So it's not so much what a gene is doing, if it's being turned on or turned off. You have to kind of know what the product of the gene is actually accomplishing to know if the action of the gene is having one of two effects on the enzymes in question. So just understand that we can use genetic control to produce products that may either slow enzyme activity down or speed enzyme activity up. Again, the one that we're really going to be focusing on is our end product inhibition. So to go back and look at a couple of terms that we already had to use, maybe we're putting the cart before the horse, but here we are again. This kind of illustrates that enzyme-substrate interactions a little bit more, and the fact that it never actually gets consumed. This would be the substrate, this would be the enzyme, and they kind of interact together physically. It's a physical interaction that helps these reactions take place. Now, this particular interaction has been described using two models. And I've had some problems with students in the past thinking that these are two different terms for the same item, and that's not the case. The two models that we're dealing with are the induced fit model and the lock and key hypothesis. Any enzyme's activity can be described using either the lock and key or the induced fit model. So we can look at any reaction taking place, any enzyme's activity, and we can describe that reaction using the lock and key hypothesis. But we can also describe that reaction using the induced fit model. Both of these look at different aspects or different characteristics of any enzyme's mechanism. The lock and key hypothesis references an enzyme's specificity. Some enzymes are very specific. There's only one substrate that they ever work on. Some enzymes are very unspecific. When we talked glycolysis, we looked at the enzyme hexokinase as it converted glucose into glucose 6-phosphate. Very specific. There's one job to do, does it well. We also talked about salivary amylase. Salivary amylase takes these large carbohydrate molecules and starts breaking them down. Very non-specific. It doesn't matter which subunits are in there. It just starts breaking it down. But whether we're looking at an enzyme that's very specific or an enzyme that's very non-specific, because we're talking about the relative specificity, we're using the lock and key model. It describes the enzyme's specificity. The induced fit model has nothing to do with specificity. It looks at how the enzyme changes shape to accommodate to its substrate. Remember, these are proteinous structures. Last time we talked about primary sequence, secondary structures, tertiary, quaternary structures. We know that that's one big chain of amino acids that can be slightly manipulated. And as we see the enzyme interact with the substrate, some enzymes undergo more conformational three-dimensional shape change than others. That is what we're looking at when we say induced fit. There's nothing wrong with looking at a scenario and describing it from two completely different contexts. We could go out into the parking lot here right now and pick a car. And we could discuss that car from two completely different viewpoints. It doesn't mean we're talking about different cars. We're still referring to the exact same car. We could talk about that car from a performance standpoint, 0 to 60 speeds, top speed, acceleration, braking, how fast it might go around a particular track. Talking about this one particular car. Someone else may look at that same car and talk about its safety, its airbags, its crash ratings, crumple zones, structural rigidity. Two completely different topics looking at the same subject. And that's what we see here. We use the lock and key model to describe an enzyme's specificity, its degree of specificity. Or we can use the induced fit model to discuss that same enzyme's ability to change shape as it interacts with the substrate. So now let's look a little bit more at some other reactions and some of the energetic models. Let's kind of remind us of the first law of thermodynamics. Energy can't be created nor destroyed, only transformed from one form to another. And let's add on our second law of thermodynamics. It's the law of increased entropy. Now, that might sound like gibberish at this point, that's fine. But when I explain it, it should make more sense. The law of increased entropy, it basically states that if left alone, things fall apart in a disorganized manner. If you've ever repetitively cleaned your house, you know what I'm talking about. If left to its own devices, things turn to shambles of disorganization. An organized state requires a continual input of energy in order to create it or maintain it. You work your tail off, getting everything in the house just the way it's supposed to be, and it seems like in 5 minutes of the kids running around, it looks like you've been robbed again. You need energy input to maintain organization. Now, if we put that into the context that we've been in, think about our proteins. You could have this jumble of loose amino acids, completely disorganized and useless to us. Or we could put energy into it and arrange them in such a way that now we got a useful protein as an enzyme, for example. If we left that enzyme alone, we would see it deteriorate back down to its spare parts again. We need to put energy into a scenario to increase its degree of organization. Now, we have two different charts. Looks very similar to the one that we had on the first slide we had, energetic diagrams. But now we're seeing them in opposition a little bit. On the left, again, energy degree up the side. We're going from a high state of energy to a low. Forget about the activation energy. Remember, it's the enzyme or the catalyst that's going to take care of that. Look at the start and the end. We start here, we end down here. Energy went from a high to a low. We call that an exergonic reaction. We call it an energy releasing reaction. If we go from high energy to a low energy, that energy had to go somewhere. Remember, it doesn't just disappear. It has to be transformed from one form to another. So in this particular reaction, maybe energy was given off as heat. It was transformed from a chemical energy into energy as heat. Maybe it was given off as light. Whatever the case may be, energy was released from that system. Over here, we go from a low energy to a high energy. And again, we can't just magically make this happen. We had to get energy from somewhere to put into that system to make up that difference. This is an energy-consuming reaction called an endergonic reaction. You're going to have to be able to look at diagrams similar to this and classify them as endergonic versus exergonic, energy-consuming versus energy-releasing. Look at the start, look at the finish, and see if energy had to be increased in that system, make it endergonic, or if energy was ultimately released from that system, making it exergonic. So the big question is, if we want to take something, these individual amino acids, and we want to put energy into the system to make a complex protein, where do we get that energy from? We get that energy from one of these reactions. The energy released from one reaction that's exergonic is going to be used to power the energy-consuming reaction that is endergonic. And that's what we mean by coupled reactions. Think back to what we talked about last time with our biochemical pathways. We went from glucose to pyruvate. 1 6-carbon molecule into 2 3-carbon molecules. We went to a more disorganized state. Energy releasing. At the same time we were going down that cascade, we were taking ADP and turning it into ATP, a higher energy molecule that needed to attach a whole new phosphate group onto the end of it to make it a more complex molecule. So we pair these reactions up in a coupled manner. One energy producing, one energy consuming, and it's the energy releasing, the energy producing reaction that's going to provide the energy input for the reaction that needs to consume it to reach the product level. So energetically, we'll always have these two reactions going in different directions, one supplying the energy for the other. So when we look at the key concepts from this section, you should understand what an enzyme is. What role does it play in the reactions we talked about last time? You should understand how we regulate enzymatic activity and what that means for the reaction of the rates of the reactions that it governs. Remember, end product inhibition was our biggie there. You should understand our two models of enzyme activity, what they describe. And finally, the concept of coupled reactions combined with endergonic and exergonic energy inputs and outputs, how they fit together to drive the energy through these systems. So if you have any questions on this, please let me know, and we shall take it from there. Thank you.